So I did my presentation on ion channels and their function in neuronal communication. So first of all, it's important to address what is an ion channel. And an ion channel is one of our classes of intermembrane proteins, meaning they span across the entire membrane of the cell membrane. And the way that they're structured, they have hydrophobic and hydrophilic amino acid side chains in their protein structure. The hydrophobic side will run through the cell membrane, as that's where the hydrophobic lipid tails are of the cell membrane, so they'll interact well there. And the hydrophilic side will be exposed to both the cytosol and the extracellular fluid. Um, and these structures create gated passage of ions. So these are not just open pathways for ions to run through in and out of the membrane, but they are gated, meaning they only will open when certain conditions are met within the cell or a certain structure binds to it. And they're permitted when they open to allow the flow of the ions down their concentration gradient, meaning it will flow from the side that there's high concentration to lower concentration. And this is an important concept in all passive transport through a cell membrane, but specifically when we're talking about a neuron, having the right ions in the right side, having the established membrane concentration is vital for the communication to be effective. So there's many different types of ion channels that we have to be aware of, because knowing when each type is active and why will help us understand the primary function that they have in communication in a neuron. So the big difference between two major types are we have voltage gated ion channels and ligand gated ion channels. So that just means that when they open is based on a different condition being met, as this image here does a good job of showing as well. Voltage gated ion channels will open or close depending on what's called the membrane potential. And the membrane potential is just the difference in ion concentration or charge difference from the outside of the membrane to the inside of the membrane. So when that difference is at a certain point, those ions will be allowed to pass through this channel. The channel will open. Compare that to a ligand gated channel, where it's not the membrane potential that matters here, it's whether or not there's the binding of the ligand or a signaling molecule to this protein channel. When the ligand binds, this channel will have a conformation change and it will open, allowing for ions to pass through it. So typically we see a ligand-gated channel, and I'll discuss this more moving forward. We'll see a ligand-gated channel used to start the signal, and typically the ligand is a neurotransmitter from the previous neuron. And then we'll see the voltage-gated channels kind of propagate the signal down the entire neuron so it can potentially be sent to the next one. When we talk neural communication, it is all just about chain reactions, connections between different neurons. So if that signal is going to be sent from one to another, the ligand, the neurotransmitter, must bind to initiate that signal on the next one. It's also important to note that these ion channels are specific for a particular ion. They don't just let any ion pass through, but generally only one can pass. And the ones that we see specifically with a neuron and its communication would be sodium, potassium, and calcium. Now, these ions are all used for different purposes in the neural communication. So when these specific ion channels are open, and understanding when and why each would be open really helps us see what the neuron is doing at that particular time. Because if our sodium channels are open, that's going to be at a different part in the communication compared to our potassium or calcium channels. So understanding when each ion channel is open will allow us to better understand how neurons actually use them to communicate. So in order to understand how neurons actually will send a signal, it's important to have a brief understanding of the parts of a neuron, because these parts are going to be referenced in the rest of the presentation. So it's good to know generally where these parts are and what they're going to do. So we have three major parts of a neuron. They are the dendrites, the soma or cell body, and the axon. So neurons tend to have multiple dendrites. Dendrites are where we receive signals from other neurons. There's all these branches here in this image, and we're going to have neurons synapsing or connecting on these. So the dendrites are receiving signals from many other neurons at the same time. And typically, this is where we see ligand-gated channels. The neurotransmitters from the previous neuron are going to be binding here to start that communication between two. The soma, this middle part here, the cell body, it integrates information from the dendrites. Since there's multiple signals hitting this neuron at the same time, we essentially have to determine whether or not that signal is going to be sent onward, sent forward. And that is all based on the change in charge, whether it's excited or inhibited. And again, that will all depend on how many ions or what type of ions are entering this neuron at the time. And the soma is kind of what determines that. And then finally, we have the axon, which is this chain thing right here. We'll talk about the different pieces of it or that covering in a few slides. But this is where if the neuron is going to send a signal, it gets sent down the axon. There's only one axon on a neuron because a neuron can only send one signal at a time. So it will send down this part here. And then you see it branch at the end to create what's called our axon terminal, where we have the little ends here to form the synapse with our next neuron, or sometimes even a muscle cell. So knowing these parts is important for understanding where the different ion channels are and what each part is going to do. And my last little overview slide here, or introductory slide, is it's important to understand very briefly the overall type of communication that is going to be used by a neuron and why we actually need ion channels to change the charge. So neural communication is what we call electrochemical communication. And breaking apart that word, there's two components to it. We have the electric component and the chemical component of our communication. So generally, the job of the neuron is to relay these electrochemical impulses, these signals all across the body. They could be basic touch in your skin, your sensory receptors, and like your fingers. Basic touch relayed to the brain. It could be sending a signal to a muscle to tell it to move. Could be controlling the release of hormones from different glands like the pituitary in the body in order to get like hormones released into the bloodstream center on the body. All of these things controlled by neural communication, and it's all the same basic thing. And that first piece is the electric signal. And I always like to explain it as electricity. If I want to send a signal from flipping a light switch to the light actually turning on, we use wires. Wires are essential because we have charged particles, energetic particles that can pass through that wire in order to relay that signal. Neurons are the same thing. They are essentially a wire. They allow for the relay, the movement of charged particles into and out of it. And that's the electric part. We call it an action potential. And the action potential is truly where we see our ion channels come into play, because the charged particles that are moving are the ions, sodium and potassium in particular. But then I always want to go to, why do we need the second part? Why do we need chemical communication in the form of a neurotransmitter? So the neurotransmitters are true molecules. They're chemicals, signaling molecules that get released from the axon of one neuron, as we can see here, and it will diffuse across what's called the synaptic cleft, the gap between one axon of one neuron and the dendrite of the next, diffuses across that synaptic cleft and binds to receptors. Typically, these receptors are found on ligand-gated ion channels, so it can start the next signal. But we need these because if you notice, these two neurons don't touch each other. So it's like a cut in the wire. And I always like to teach that electricity doesn't jump. You can't cut a wire and expect the signal to still get from one place to the next. So because this wire, this neuron is cut, we need a chemical to bridge that gap. So the electric signal goes through one neuron. The ions go in and out of that neuron to relay a signal to the end. Once it reaches the end, we will get a release of a neurotransmitter, the chemical component, to pass across the synaptic cleft and start the signal again. So getting into our different pieces here, we want to look at what an action potential is. I mentioned that that is our electric communication. It is the primary process of neural communication. Without an action potential, that means the neuron is not sending a signal. So the action potential involves strategic opening and closing of different ion channels. Keyword here is strategic. It's going to open certain channels at certain times in order to change the membrane potential. And that's what's shown in this graph here. So our membrane potential is always sitting at negative 70 millivolts at its resting state. So we call that the resting potential. And that doesn't mean that the charge is measured inside the neuron and that it's negative 70 millivolts, but it's a comparison. It's 70 millivolts less on the inside than it is on the outside of the cell. And we change that by allowing our ions in. If we think back a couple slides ago to the major types of ion channels that we see, we have sodium, potassium, and calcium. All three of those ions have positive charges. So if we're letting these ions in, we will see an increase in their charge, and we call that depolarization. Now, we like to say that because there's a potential, there's a membrane potential, the membrane is polarized, meaning it has different sides, just like north and south pole of a magnet. They're not the same. So one side of the cell, one side of the membrane is a different charge than the other. When we depolarize it, we are getting rid of that difference. We actually cross zero and go into around positive 40 millivolts. So we're getting a rush of positive ions into the cell. And that is our depolarization step. It's our action potential. Then when we're repolarizing, we're bringing it back down to our resting state. And that is the removal of positively charged ions to bring our charge back down. So when we open and close certain ion channels, we get this movement, and that's what this graph is showing. And it's all the result of a stimulus to create the action potential. Once enough stimuli have been detected by the dendrites, we cross that threshold point here, and our action potential can begin. And lastly, the changes in our potential allow for our signal to propagate down the axon. So we use a change in charge to kind of go stepwise down our axon until we reach the end. So that's how we're able to communicate from one end of the neuron, the dendrites, to the other down the axon. So breaking it apart piece by piece here, we first have the signal reception component, and that takes place at the dendrites. And our dendrites contain ligand-gated ion channels. And the neurotransmitters, like I mentioned before from the previous neuron, are the ligand. When we get a signal that the dendrites pick up, it can either be excitatory or inhibitory. And whether it's excitatory or inhibitory just means, is it going to send the signal onward, or is it going to shut this neuron down? And what determines that is whether or not it's a positive ion or negative ion that is allowed to enter. And that's all based on the neurotransmitter that ends up being attached. Different neurotransmitters will create different results in our receiving neuron, in our postsynaptic neuron, which this would be. If it's an excitatory signal, typically we're going to allow positive ions into this neuron. A lot of times we get ligand-gated sodium channels that open, allowing sodium to enter. One thing that is important to note here, whenever we get sodium channels opening, we will see the influx of sodium into our neuron because the concentration gradient has significantly more sodium outside the neuron than in. And when we see potassium channels opening, we will see those ions leaving the neuron, as we have a lot more potassium built up inside the neuron than out. So that's how we're able to use concentration gradients in order to move ions through passive transport the way that we should. When we get an inhibitory neurotransmitter that binds to the dendrite, it usually brings the neuron further away from that threshold point, so we're making it more negative. Typically, an ion that enters when we get an inhibitory stimulus would be like chloride ions, which have a -1 charge. So that brings our membrane potential further from threshold, slowing it down. And that signal will continue down the neuron if threshold is reached, based on the integration done in the cell body. So these neurons, like I mentioned previously, are receiving many signals at one time. So we get positive signals, excitatory, we get inhibitory signals. And when we add that all together, if threshold is reached, then the signal will continue down the axon. If threshold is reached, we will start the signal continuation here, which would begin with depolarization of the axon of the neuron. So when threshold is reached, we get voltage-gated ion channels opening, specifically voltage-gated sodium channels. And this, again, goes back to that voltage-gated. Threshold is the signal to open our next ions. If we don't reach this point right here on the graph, that -55 millivolts, we will not see these ion channels start to open. If they don't start to open, we will never see this signal being sent. We'll never see that positive influx of sodium ions into the neuron. So that's what we see, these little stimuli here, these failed initiations. They're excitatory signals, but it's not enough to get threshold to be reached. Once it happens, those voltage-gated sodium channels begin to open, beginning at the soma, so beginning at the furthest end of the axon away from the end. Once that sodium channel or the sodium channels begin to open, you will see a flow down the axon. They'll begin to open little by little down that axon, allowing it to propagate. So when we get the influx of sodium ions in one spot, it signals the next cluster of voltage-gated sodium channels to open, allowing more sodium to come in, and so on. That signal continues and gets magnified in a matter of a millisecond or so, creating that depolarization, creating that action potential, getting all the way down to the end of the wire, or the axon terminal, the very end of the signal. One thing that's important to note for how these signals are actually sent is there are structures on our axon called neuroglia, or glial cells. Specifically, this type of glial cell is a Schwann cell, creating what's called the myelin sheath. And the myelin sheath, it's basically insulation wrapped around our axon, and it creates concentrated pockets of sodium channels. So those concentrated pockets are in what we call the nodes of Ranvier, which are gaps of our myelin sheath. And this allows our signal to jump down our neuron. Instead of having to go like one little piece by piece, it depolarizes the pocket, and then it sends the next one and the next one. So it's a lot faster. These two axons show kind of a comparison here. In this axon, our signal is able to jump from here straight to here and so on, all the way down, just kind of skipping along. And in this one, it would have to go kind of inch by inch here, allowing each part to separately depolarize. It just takes a lot longer. It's an uninsulated signal. So I like to teach this using an example of getting an expo marker from one end of the room to the other, using a chain of students, and one chain has to pass it individually to each student one by one, and the other line can throw it stepwise. And obviously the line that can throw the marker will get it to the end faster because you can skip steps. So it creates clusters of our ion channels. Now, once we've depolarized, we have basically broken our membrane potential here. We've created the opposite effect. We have to bring our membrane potential back down. And this will involve strategic closing and opening of our ion channels again. So we want to close our voltage-gated sodium channels. This will stop the influx of positive ions, kind of reaches our pinnacle point here. And then we end up opening our voltage-gated potassium channels. When potassium channels are open, potassium will flow out, and that will bring our charge back down. We call it repolarization, and it will bring our membrane potential back to normal. So sodium channels, voltage-gated sodium channels allow sodium in, voltage-gated potassium channels allow potassium out. Now, yes, our membrane potential is reset, but our ions are flipped. We still want to make sure we have a concentration gradient here so we can do this signal again in a matter of milliseconds here. So we use what's called the sodium-potassium pump to reset our concentration gradient. It will pump sodium out of the cell, it will pump potassium back in, allowing us to reset our ions and get us back to our resting state here so our neuron is able to send the next signal. The final step in our process here is that we need to actually release the neurotransmitter. And this involves a third type of ion channel, specifically calcium channels. So when that electric signal reached the end of the axon, we got to get that neurotransmitter released in order to get the signal to the next neuron. And like I mentioned previously, electricity can't cross a cut wire. So we have to release this neurotransmitter, this chemical, but it has to be at the right time. If we release neurotransmitters when we're not supposed to, when the signal isn't actually there, it'll cause all sorts of problems in our neural communication. It could cause spontaneous muscle contraction, feeling things that aren't actually happening. So it's important to get a signal correct here. And when we reach that signal, it opens voltage-gated calcium channels at the end. Calcium entering the neuron is essential for neurotransmitter release. It creates a condition where our vesicles in our synapse, our synaptic vesicles containing the neurotransmitters, are able to merge with our cell membrane. When that happens, it releases our neurotransmitter into our synaptic cleft, allowing it to diffuse across to the next neuron. So calcium allows this fusion to take place that we see here between our vesicle and our membrane, allowing for neurotransmitter release. Without voltage-gated calcium channels opening, allowing that calcium in, we would not see our neurotransmitters being released. So just want to quick summarize our mechanism of action here for our communication. We get neurotransmitters that bind to receptors on the dendrites, and this will open ligand-gated ion channels, with the neurotransmitter acting as the ligand. If it's excitatory, you'll see sodium generally, and if it's inhibitory, you'll see chloride. If we get enough excitatory signals, if threshold is reached, the signal is going to be relayed. It is then sent down the axon. Our voltage-gated sodium channels will open, and that signal will propagate down the axon, creating that depolarization. And then once that is complete, our voltage-gated potassium channels will open, and it allows for potassium to leave. We close those sodium channels up as well. Potassium can leave and bring our membrane potential back to normal. And this signal causes voltage-gated calcium channels to open at our synapse, allowing for the neurotransmitter to be released. And this entire process can restart at the next neuron. I just want to mention a couple things that could potentially impact our neural communication through our ion channels. So sometimes our ion channels can be blocked. And there's a few different toxins that actually will block our ion channels, which in turn blocks our signaling. So we get our what's called the dendrotoxin, and this is a toxin commonly found in poisonous snakes, and it blocks our voltage-gated potassium channels. And if our potassium channels are blocked, we never can bring our neuron back down. It kind of paralyzes the neuron and doesn't allow for our signal to ever be sent and complete. We also see tetrodotoxin, which is found in pufferfish, among other things, and this blocks our voltage-gated sodium channels. So it prevents our signal from ever being sent from one neuron to the next. Even if the neuron is being told it needs to create a signal, it just can't do it. If voltage-gated sodium channels don't open, we cannot get that signal propagation down our axon. And this graph, these two graphs on the bottom here, show the result of tetrodotoxin in a neuron. So this first one, we see normal conditions. We see the post-synaptic response, meaning the depolarization of the second neuron here based on the signal being sent. And here we see that same neuron, but with the presence of tetrodotoxin, you'll see that it is not able to send a signal because it's being blocked by this toxin. You can't get an action potential. And then a clinical use is lidocaine. So our topical or sometimes injected numbing agent used as pain relief, a lot of times used at a form of it used at the dentist. Lidocaine blocks sodium channels locally, so wherever we put it. And if our pain receptors have their sodium channels blocked, we are unable to feel the pain. It's not able to send the pain signal. It's also why if you've ever had this topical numbing, you can't really move that area very well. You can't feel anything there because our neurons are essentially paralyzed through the blocking of these channels. And then there is a couple diseases that are associated with ion channels. I want to talk about one. We have myasthenia gravis. And this is the blocking of not a voltage-gated channel, but a ligand-gated channel. It blocks our acetylcholine receptors on our muscle cells. So muscles undergo action potentials just like neurons in order to create a contraction. It has to get a ligand binding, opens a ligand-gated sodium channel, and then that starts the voltage-gated signaling to get the muscle to contract. So when people have myasthenia gravis, the ligand-gated sodium channels are rendered inactive. So these ion channels are not able to bind with acetylcholine, which is the neurotransmitter that tells a muscle it is time to create a contraction. So that blocking will create paralysis of the muscle. And this can be sometimes a local thing, or it can be across larger areas of the body where muscles are just not able to get a contraction. And this can, like I said, create partial paralysis, oftentimes extreme muscle weakness and fatigue through not enough muscle cells getting the contraction signal. sometimes it does create full-on paralysis due to lack of binding of our ligand-gated channels. So, in summary, both our ligand-gated channels and our voltage-gated channels are essential for our neurons sending a signal and for these signals being received from both our sending and receiving neurons. And here are the references that I used for this presentation.